Welcome to the Deep Dive, the show that unlocks the secrets hidden within the very blueprint of life. Today, we're embarking on a truly profound journey into genomes and their evolution, drawing our insights from a deep dive into the latest scientific understanding, particularly from detailed biological sources like Campbell Biology and Focus.
That's right.
Our mission, as always, is to take some of the most complex biological information and distill it down into core insights, making these dense concepts clear and relevant for you, even, without visuals. We're going to reveal some surprising facts about our own genetic makeup and, crucially, how the incredible diversity of life on Earth actually came to be. Think of it as a master key to understanding life's fundamental operating system.
Absolutely. It's fundamental.
To really set the stage for this deep dive, let's start with a couple of fascinating extremes highlighted in our sources. So imagine the elephant shark, right? An ancient creature often called a living fossil. Its genome holds the record for the slowest evolving among all vertebrates sequenced so far.
Wow. Slowest evolving.
Yeah. Now, at the other end of the spectrum, you have the tiger tail seahorse, whose genome is the fastest evolving of all known fish genomes.
Such a contrast.
Exactly. These two wildly different rates of change pose a big question. What can these extremes, and everything in between, tell us about the grand, ongoing story of evolution?
Well, what's truly remarkable here is how these divergent paths highlight the sheer volume of genetic data scientists can now collect and, importantly, actively analyze. This explosion of information has birthed entirely new fields like genomics, that's the study of entire sets of genes and their interactions, and bioinformatics, which applies powerful computational methods to store and interpret all this biological data.
Right.
We've moved far beyond just looking at simple lists of A's, T's, Z's, and G's. These are now, powerful tools for deciphering life's deepest mysteries.
That explosion of data is incredible. But once you have all those billions of letters, the real challenge begins. How do you actually make sense of it? This ability to collect and interpret vast amounts of genetic data truly marks a foundational shift in biology. And it really kicked off with an ambitious undertaking known as the Human Genome Project, which aimed to map out the entire sequence of our genetic code.
A monumental task.
Absolutely. The technological leaps that came out of the Human Genome Project were nothing short of revolutionary. Picture this. In the 1980s, a good lab could sequence maybe 1,000 base pairs a day.
Just 1,000.
Right. Fast forward to just a few years ago, and automated machines could sequence tens of millions of base pairs per second.
Per second. That's mind-boggling.
And the cost. Sequencing the very first human genome took 13 years and cost roughly $100 million. Today, you can get a person's entire genome sequenced in a day or less for about $1,000. That's not just faster and cheaper, it's a fundamental democratization of access to our genetic blueprint.
That dramatic reduction in costs and increase in speed was driven by continuous innovation in techniques. The core method often used, still refined today, is called the whole genome shotgun approach. Imagine taking all the DNA from an organism, randomly chopping it into millions of tiny overlapping fragments,
Okay.
and then sequencing each of those short pieces. Then powerful computer programs take those fragments and meticulously assemble them like a giant, incredibly complex jigsaw puzzle into a single continuous sequence. The computational power required for this kind of assembly is absolutely staggering and crucial to the entire process.
And even that process has evolved. Newer techniques like sequencing by synthesis have further accelerated things and slashed costs by eliminating some of those initial time-consuming steps, allowing for many small fragments to be sequenced all at once. It's a constant race to make it faster, cheaper, and more accessible.
Yeah, the tech just keeps improving. And what's also fascinating is how these advances allow us to look beyond individual organisms. Consider metagenomics, where scientists collect DNA directly from an entire community of species within an environmental sample, say, from the complex microbial world inside a cow's rumen where hay is digested.
Right, like a whole ecosystem's DNA.
Exactly. In a breakthrough in 2018, researchers sequenced DNA from cow rumens and managed to identify the genomes of over 900 microbial species. And get this, over 60,000 previously unknown genes involved in digesting carbohydrates.
60,000, wow.
The huge advantage here is that it allows us to study microorganisms that are very difficult or even impossible to grow and study separately in a lab. You get the whole picture.
So we've talked about generating all this data from individual genomes to entire ecosystems, but once you have literally billions of genetic letters, how do scientists actually make sense of it? This is where bioinformatics truly steps in as the crucial field that applies computational methods to store, organize, and analyze all that raw biological data.
Precisely. And the world relies on centralized resources that make this vast ocean of data accessible globally. For instance, the National Center for Biotechnology Information, or NCBI website, hosts the GenBank database, which, as of 2018, contains sequences from hundreds of millions of genomic DNA fragments, totaling hundreds of billions of base pairs. And it's constantly expanding.
Just an unbelievable amount of information.
It really is. And tools like BLAST, a widely used software program, are like super fast genetic search engines. You can compare a new DNA sequence with every other sequence in GenBank, base by base, to find similar regions within or between species. There are also programs for comparing protein sequences and even visualizing complex 3D protein structures.
That's incredible. But how do scientists actually use this data to figure out what a gene does? If you discover a new sequence, what's the next step?
Well, often they deduce its likely function by comparing it to known genes or proteins from other organisms.
Mm.
It's frequently more informative to compare protein sequences rather than DNA, since multiple DNA sequences can actually code for the same protein.
Okay, that makes sense.
And then, they might confirm if that gene is even active, perhaps using a method like RNA sequencing, often called RNA-seq.
Right. See if it's actually being used.
Exactly. But this brings up an important challenge. What happens when a sequence only partially matches a known gene? Or even more difficult, when it's entirely new. Like about a third of E. coli genes were completely unknown when its genome was first sequenced.
So what do you do then?
In those situations, a protein's function is typically deduced through a combination of biochemical and functional studies. This might involve determining the protein's complex 3D shape, or critically knocking out or disabling the gene in an organism, maybe using powerful molecular tools like CRISPR-Cas9.
Ah, the gene editor.
Right, which acts like a precise genetic scissor to observe the resulting effects on the organism. You see what breaks when the gene is gone.
And as we get better at analyzing individual genes and proteins, the scientific focus is really shifting to understanding them at a systems level. This is where genomics, the study of entire sets of genes, and proteomics, the study of entire sets of proteins, truly come into their own.
Absolutely. The ultimate goal here is systems biology, which aims to build dynamic models of entire biological systems by studying the complex interactions among all their parts. The ENCODE project, a massive research effort on the human genome, is a prime example. Its aim was to identify all the functional elements in our genome.
And what did they find?
Well, the most striking finding was that the vast majority of our DNA, once dismissively called junk DNA,
Right, I remember that term.
is actually a bustling control center. About 75% is transcribed into RNA at some point, and a significant portion has assigned biochemical functions, even though less than 2% of it actually codes for proteins. This truly changed our understanding of how our DNA works. It's not junk at all.
That's a huge shift. And this systems biology approach has direct, life-changing applications in medicine, right? Like with the Cancer Genome Atlas project.
Exactly. That project analyzes how many interacting genes and gene products behave together to understand how changes in these biological systems lead to cancer. By comparing gene sequences and expression patterns in cancer cells versus normal cells, scientists are identifying common mutations and, crucially, potential new drug targets for various cancers, including those aggressive metastatic tumors that are so challenging to treat.
So it's helping find new ways to fight cancer.
Yes. And if we connect this to the bigger picture, it leads us directly to the burgeoning field of personalized medicine. Using advanced techniques to analyze gene expression patterns in individual patients allows physicians to tailor treatments specifically to that patient's unique genetic makeup and the specific characteristics of their cancer. Breast cancer treatment is a fantastic example where this personalized approach is already making a real impact.
Tailoring the treatment to the person.
Precisely. In the not-too-distant future, our medical records may even include our own unique genetic barcode, offering incredible potential for disease prevention and truly customized treatment.
That's incredible potential. So we've explored the revolution in how we sequence and analyze genomes, but now let's turn our attention to the genomes themselves. They vary wildly in size, the number of genes they contain, and how densely packed those genes are across the incredible diversity of life.
That's right. If we look at general trends, bacterial and archaeal genomes are typically quite compact, usually just a few million base pairs long.
Relatively small.
Yeah. Eukaryotes, however, tend to have much larger genomes. Yeast, a single-celled fungus, is around 12 million base pairs, while most multicellular animals and plants have at least 100 million. Humans, for example, have about 3 billion base pairs. But here's where it gets interesting. There's a surprising lack of clear correlation between genome size and organismal complexity.
Oh, so bigger doesn't always mean more complex.
Not necessarily. For instance, a cricket genome can have 11 times more DNA than a fruit fly, and some plants have genomes that are truly gigantic, dwarfing our own. There's this one Japanese flower, Paris japonica, with a genome nearly 50 times larger than ours.
50 times. That's wild. And the number of genes also varies widely, right?
It does. Prokaryotes typically have a few thousand genes. Eukaryotes, on the other hand, range from about 5,000 up to tens of thousands in some multicellular organisms. But here's where it gets really surprising. One of the most unexpected findings from the Human Genome Project was that humans have fewer than 21,000 genes.
Wait, fewer than 21,000, that seems low.
It's similar to a tiny worm like C. elegans, and much, much lower than the initial estimates that were in the range of 50,000 to 100,000 genes.
So how do we manage all complexity with, well, relatively few genes?
Yeah, that's the fascinating question. How do humans get more bang for the buck with fewer genes than initially expected? A major factor is extensive alternative splicing of RNA transcripts. Think of it like a single recipe that, depending on how you read and interpret it, can create 10 different dishes.
Ah, okay. One gene, multiple proteins.
Exactly. Our bodies do this with genes. Over 90% of our multi-exon genes are spliced in at least two ways, generating multiple different protein products from just one genetic construction. Beyond that, there are additional ways to diversify proteins after they're made, through post-translational modifications, and the crucial regulatory roles of small RNAs also play a big part.
So it's not just about the raw number of genes, but how cleverly we use them. This brings us to gene density, the number of genes per given length of DNA.
Right, and here again, there's a big difference. Prokaryotes have a much higher gene density. Most of their DNA consists of genes, packed pretty tightly. But in eukaryotes, especially mammals like us, gene density is significantly lower. In fact, we have vastly more non-coding DNA than bacteria.
That non-coding DNA, that's the stuff that used to be called junk DNA.
That's the one. In humans, the vast majority of our DNA, like 98.5%,
Yeah.
doesn't code for proteins.
Mm.
For a long time, it was dismissively called junk DNA.
Yeah.
But recent large-scale research projects like ENCODE have strongly suggested that these non-coding regions are far from junk.
So what is it doing?
Well, they're actually highly conserved across species, which usually means they're important. They seem to play crucial functional roles in regulating our genes, even if we're still uncovering all their specific jobs. This includes segments within genes called introns, which get spliced out,
Okay.
and also pseudogenes, which are like old, broken copies of genes that no longer function. But the largest chunk is made up of what's broadly called repetitive DNA.
A repetitive DNA. Okay, let's talk about that. What about transposable elements, often called jumping genes? That sounds pretty wild.
It is pretty wild. These are segments of DNA that can literally move from one location to another within the genome. The first evidence for these wandering DNA segments came from the pioneering work of the American geneticist Barbara McClintock. She observed changes in corn kernel color back in the 1940s and 50s.
In corn.
Yeah, in corn. She bravely proposed these mobile genetic elements, a radical idea at the time that was initially met with skepticism. But her careful work was validated years later, and she eventually received the Nobel Prize in 1983. A true trailblazer indeed.
That's an amazing story. So what are these jumping genes doing?
Her discovery was groundbreaking. There are basically two main types. Transposons move as DNA segments, either by a cut-and-paste mechanism or a copy-and-paste one. The more prevalent type in eukaryotes are retrotransposons. These move via an RNA intermediate.
RNA, how does that work?
They always make an RNA copy of themselves and then use a special enzyme called reverse transcriptase to turn that RNA copy back into DNA, which is then inserted into a new location in the genome. Interestingly, this enzyme is also found in retroviruses, like HIV, suggesting a potential evolutionary link.
Huh.
And these retrotransposons can make up a significant portion of genomes, anywhere from, say, a quarter to half of mammalian genomes and a staggering 85% of the corn genome.
85%, just from these mobile elements.
Yep, in corn. It's incredible. And we have some significant families of these jumping genes in humans, too. Allo elements make up about 10% of our genome. They're relatively short, don't code for protein, but many are transcribed into RNA and are thought to help regulate gene expression.
Okay, 10% is a lot.
It is. And even larger are line 1 or L1 elements, which count for 17% of the human genome. These are longer, and while they typically have a very low rate of transposition, they don't jump around that often. What's truly interesting is that L1 retrotransposons have been found to be more active in the developing brain.
In the brain, what are they doing there?
Well, it's thought they might potentially contribute to neuronal diversity and function, maybe making brain cells slightly different from each other. It's an active area of research.
Fascinating. Are there other kinds of repetitive DNA besides these jumping genes?
Oh, yes. Beyond transposable elements, other forms of repetitive DNA account for a significant portion of the human genome. A notable type is simple sequence DNA, which consists of many copies of short sequences repeated over and over again, like GT-Tech, GT-Tech, GT.
Go repeats.
Exactly. When the repeating unit is very short, just 2 to 5 nucleotides, it's called a short tandem repeat, or STR. The number of these repeats varies greatly from person to person, and this variation is incredibly useful.
How so?
It's used in STR analysis for genetic profiling and forensics, like DNA fingerprinting. It helps identify individuals or even exonerate wrongly convicted people. The Innocence Project has used this extensively. These simple sequence DNAs also play crucial structural roles at the ends of our chromosomes, called telomeres, and in the constricted region, the centromere.
Wow, so even simple repeats have vital jobs. So when we put it all together, it's quite amazing. Only about 1.5% of the human genome actually codes for proteins.
Just 1.5%.
Right. But if you include those introns and regulatory sequences, roughly a quarter of our genome is considered gene-related. It's a remarkably complex and efficient system in its own way.
It really is. And within that gene-related DNA, we find multi-gene families. These are collections of 2 or more genes that are either identical or very similar.
Like backups or variations on a theme.
Both, kind of. A classic example of identical genes are the hundreds to thousands of copies of rRNA genes. These code for ribosomal RNA, which is essential for building ribosomes, the cell's protein factories. You need millions of ribosomes, so having many copies of the RNA genes allows for rapid production.
Makes sense. And non-identical ones.
For non-identical genes, the globin gene families are a perfect illustration. We have alpha globin genes and beta globin genes located on different chromosomes. Different versions of these are expressed at different developmental stages, like in embryos, fetuses, and adults.
Why different versions?
They allow for hemoglobin molecules with different oxygen affinities, which is crucial for efficiently transporting oxygen from mother to fetus, for example, and then adapting after birth. These families also contain pseudogenes, which are like former genes that have accumulated mutations and lost their function over evolutionary time. They're like molecular fossils in our genome.
Molecular fossils, I like that. This brings us to the exciting part. How do genomes actually evolve? At the simplest level, it starts with mutation, right? Small changes in the DNA sequence.
Right, mutation is the ultimate source of variation, but the changes can be much, much grander than just single letter swaps. Large-scale changes include phenomena like polyploidy. Polyploidy. It's where an organism ends up with extra full sets of chromosomes, often due to errors in meiosis, the cell division that makes sperm and eggs. While it's rare in animals, it's quite common in plants and can be a major driver of plant evolution. It can lead to new species because one set of genes continues to provide essential functions while the extra set is free to diverge and potentially acquire entirely new roles.
So getting whole extra sets of chromosomes and the structure of individual chromosomes can change too.
Absolutely. Chromosomes can break and the pieces can get rearranged. They can be deleted, duplicated, inverted, or even fused together or swapped between different chromosomes. Our own human chromosome 2 is a fascinating case.
What's special about it?
It's actually the result of the fusion of 2 separate chromosomes that are still found as distinct chromosomes in our closest relatives, like chimpanzees. The evidence is really strong. You can find sequences that look like telomeres, which normally cap the ends of chromosomes buried in the middle of our chromosome 2, right where the fusion likely happened. And there's evidence of an extra inactive centromere, too.
So our genome literally shows evidence of ancient chromosomal mergers.
Exactly. And what's truly illuminating is how comparative genomics helps us trace these rearrangements across species. Comparisons between human and mouse chromosomes, for instance, show large conserved blocks of genes, meaning those groupings have remained together over millions of years, even if the chromosomes themselves got shuffled around.
So genes tend to stick together in blocks.
Often, yes. And studies suggest there might have been an accelerating rate of these kinds of inversions and duplications in mammalian evolution around 100 million years ago. This period of increased genomic shuffling might have contributed to the rapid emergence of new species, perhaps by causing problems during meiosis in hybrids, reproductively isolating populations.
Okay, so large-scale changes. What about smaller duplications, like just one gene getting copied?
That happens too, and it's incredibly important. Gene-size duplications can occur through mechanisms like unequal crossing over during meiosis, often facilitated by those transposable elements we talked about, as they can provide similar sequences for chromosomes to mistakenly align with. Or sometimes it happens through slippage during DNA replication.
Bitch.
Yeah, where the replication machinery kind of stutters and accidentally copies a short stretch twice. These errors can lead to gene deletions or duplications, and they're thought to be a major driving force behind the existence of those multi-gene families we discussed, like the globins.
Ah, so that's how you get multiple copies to begin with.
Precisely. And this is where we can really see the evolution of genes with related functions in action. The human globin genes are a textbook example. The current model suggests all globins evolved from a single ancestral globin gene that duplicated maybe 450 to 500 million years ago.
Wow, 500 million years ago.
Right. That duplication allowed the two copies to diverge, eventually becoming the ancestors of the separate alpha and beta globin gene families we see today, located on different chromosomes. Further duplications within each family, followed by mutations, led to the diverse set of globin genes we have now, those different versions expressed at different life stages.
And natural selection kept the useful one.
Exactly. Natural selection maintained the beneficial changes, like those different oxygen affinities, which are crucial for adapting to different environments, like inside the womb versus breathing air. Even the presence of those inactive pseudogenes within these families serves as compelling evidence for this long evolutionary history of duplication and divergence.
That makes sense. But sometimes a duplicated gene copy takes on a completely new role, right? Not just a variation on the old theme.
Yes, that's the evolution of genes with novel functions. A really compelling example is the relationship between lysozyme and alpha-lactalbumin. Lysozyme is an enzyme found in animals that helps break down bacterial cell walls as part of our defense system.
Okay, defensive enzyme.
Alpha-lactalbumin, on the other hand, is a non-enzymatic protein that's essential for milk production in mammals. Structurally, they're quite similar. And it turns out that the lysozyme gene duplicated in an early mammal ancestor, but not in birds, for instance. One copy of that duplicated lysozyme gene then evolved over time into alpha-lactalbumin.
So a defense gene evolved into a milk protein gene?
That's the idea. It directly links a gene duplication event to a key mammalian characteristic, the ability to produce milk. A totally new function emerged from an old gene.
That's incredible. What about pieces of genes evolving, like exons?
Yeah, that's another fascinating layer. Exon duplication and exon shuffling. Remember, proteins often have modular structures made of discrete functional regions called domains, and these domains are frequently encoded by individual exons, the coding segments within a gene.
Right, the exons are the bits that code for protein.
Correct. So unequal crossing over can sometimes duplicate just a single exon, or maybe a few exons, within a gene. This can potentially augment the protein's function by, say, adding another binding site. Exon shuffling is even more dynamic. It's the mixing and matching of different exons, perhaps from completely different genes, usually due to errors during meiosis that cause breaks and rejoining within the intron regions.
So you can literally shuffle functional modules between genes.
Exactly. This incredible process can create entirely new proteins with novel combinations of functions. A great example is the tissue plasminogen activator, or TPA protein. It's involved in dissolving blood clots. It's thought to have evolved relatively recently through the shuffling of exons borrowed from other unrelated genes coding for different protein domains, like building with molecular Lego bricks.
Molecular Lego, I like that analogy, and it seems those jumping genes, the transposable elements, pop up again here.
They absolutely do. It's clear that transposable elements play a truly significant and dynamic role in genome evolution. They can promote recombination by providing similar sequences in different locations, sometimes even between non-homologous chromosomes. They can disrupt cellular genes or their control elements, just as Barbara McClintock observed in corn, changing kernel color.
Right.
And they can even carry entire genes or individual exons along with them when they move, effectively transplanting genetic material to new locations. Now, while most of these effects are likely harmful to an organism,
Because they're messing things up.
yeah, disrupting a working gene is usually bad news.
Hmm.
But the rare beneficial changes a new regulation pattern. A novel gene fusion provide the essential raw material for natural selection to act upon. They generate variation, leading to incredible genetic diversity over vast stretches of evolutionary time.
So after diving into the intricate mechanics of genomes and how they change, let's shift our focus slightly to how comparing genomes provides powerful clues to both evolution and development, truly bridging the macro and micro levels of biology.
Right, and the underlying principle is beautifully straightforward. The more similar the sequences of genes and entire genomes are between two species, the less time has passed since they diverged from a common ancestor. So more similarity means they're more closely related.
Makes sense.
Comparing very distantly related species, like the three domains of life, bacteria, archaea, and eukaryotes, which diverged billions of years ago, helps us clarify ancient evolutionary history. For example, it was really surprising finding that nearly half of human genes could actually replace important genes in yeast, a single-celled eukaryote. That underscores our deep shared evolutionary origin with even such a seemingly simple organism.
Half our genes work in yeast. That's amazing.
It highlights the conservation of fundamental life processes. Then, comparing closely related species sheds light on more recent evolutionary events and helps us directly link specific genetic differences to observable physical or behavioral differences between those species.
And perhaps the most compelling example of comparing closely related species is the human-chimpanzee comparison. We diverged only about 6 million years ago, which is relatively recent in evolutionary terms.
Right.
While our genomes differ by only a tiny percentage, about 1.2% in single nucleotide changes, or SNPs, there's a more significant difference, maybe 2.7%, due to larger insertions or deletions of DNA segments. This includes human-specific duplications, some of which have been linked to various diseases, and even more ALU elements, those jumping genes, in the human genome compared to chimps.
And adding the bonobo genome to the comparison has provided even finer detail. It actually showed that sometimes human sequences are more closely related to either chimpanzee or bonobo sequences than chimps and bonobos are to each other in certain regions. It allows for an even more nuanced reconstruction of our recent evolutionary history.
So it's not a simple branching, it's more complex.
Exactly. What's also intriguing is looking at which genes appear to be evolving fastest in the human lineage compared to chimpanzees and mice. These include genes involved in defense against diseases like malaria and tuberculosis, suggesting pathogens have been a strong selective force. Also, genes related to brain size and development show rapid change, and notably, transcription factors, those master switches that regulate other genes.
And one transcription factor that truly stands out is the FOXP2 gene. You hear a lot about this one.
You do, and for good reason. This gene shows rapid evolutionary change, specifically in the human lineage, and is strongly linked to vocalization and speech. The evidence is really compelling.
Like what?
Well, human mutations in this gene cause severe speech and language impairment. It's actively expressed in the brains of songbirds when they're learning their complex songs. And knockout experiments in mice, where the FOXP2 gene was disrupted, resulted in malformed brains and a failure to vocalize normally as pups.
Wow. So it seems crucial for complex vocal learning and production.
It certainly seems to play a key role. And what's truly astonishing is that in 2014, scientists achieved a high-quality sequence of the Neanderthal genome.
Neanderthals, what did they find?
It revealed that their FOXP2 gene is identical to the modern human version.
Identical, so.
So it suggests that Neanderthals likely had the genetic capacity for speech, similar to us. This prompted a major reevaluation of our image of our extinct relatives, highlighting how genetic data can dramatically reshape our understanding of the past. It also reinforces the immense value of model organisms, mice, fruit flies, tiny worms like C. elegans, which are invaluable for studying complex human disorders like Parkinson's, alcoholism, and aging, precisely because so many of our fundamental genes like FOXP2 are deeply conserved across species due to shared ancestry.
Right, we can learn about ourselves by studying them. Beyond species comparison, scientists are also comparing genomes within a single species, focusing on human genetic variation.
Yes, because we're not all identical, obviously. The most common variations are those single nucleotide polymorphisms, or SNPs, tiny single base pair differences that occur frequently throughout the human genome, maybe one every few hundred base pairs.
So mostly small differences.
Mostly small, but there's also the widespread occurrence of copy number variants, or CNVs. These are larger duplications or deletions of longer stretches of DNA, maybe thousands or even millions of base pairs. These CNVs tend to have greater effects on our physical characteristics, our phenotype, and they really blur the idea of a single normal human genome. There's a lot of structural variation between perfectly healthy people.
So normal is actually quite variable.
Very much so. And these CNVs, SNPs, and variations in repetitive DNA like STRs are incredibly useful as genetic markers for studying human evolution and ancient migratory routes. For instance, comparing the genomes of individuals from diverse African communities, like the Khoisan peoples of southern Africa, revealed incredible genetic diversity. Some Khoisan individuals differ more genetically from each other than, say, a European would from an Asian.
Wow, that much diversity within one group.
It highlights the deep genetic roots and immense diversity within African populations, reflecting the fact that modern humans originated in Africa and have lived there the longest. It provides fascinating clues about ancient human migrations across the globe.
Okay, and finally, let's bring it all together with evolutionary developmental biology, often called evo-devo. This sounds like linking evolution directly to how we grow.
That's exactly what it is. This exciting field compares the developmental processes across various multicellular organisms to understand how these processes themselves have evolved, and, crucially, how subtle changes in development can lead to the incredible diversity of body forms we see around us.
So how development changes evolution and vice versa.
Right. And at the heart of EvoDevo are homeotic genes. These are master regulatory genes that control the overall body plan, determining things like where wings or legs or antennae should develop on an insect, or the arrangement of vertebrae in a mammal. Many of these genes contain a highly conserved 180 nucleotide sequence called the homeobox.
Homeobox, I've heard of that.
Yeah, it's famous because it's so incredibly conserved. This homeobox codes for a specific 60 amino acid protein segment called a homeodomain, which functions as a transcription factor. It binds to DNA and regulates entire batteries of other developmental genes, essentially orchestrating the body's pattern formation during embryonic development.
And it's conserved across many species.
Amazingly so, it's found across incredibly diverse invertebrates, vertebrates, and even in plants and yeasts. It often maintains the same relative order on the chromosome in different animal groups. This suggests this homeobox DNA sequence evolved very early in the history of life, probably in an ancestor common to all these groups, and was so fundamental and beneficial that it's been conserved virtually unchanged for hundreds of millions of years.
So this leads to the big question. How can these same highly conserved genes be involved in developing such vastly different animals, like a fruit fly, a mouse, or even us? Our body plans are so different.
That's the core puzzle EvoDevo tackles. And the answer lies not necessarily in big changes to the coding sequences of the homeotic genes themselves, but often in surprisingly small changes in their regulatory sequences, the bits of DNA that control when and where these genes are turned on or off during development.
So it's about the timing and location of gene activity.
Exactly. These subtle shifts in expression patterns can lead to major changes in body form. For example, differing patterns of how certain Hox genes, a major class of homeotic genes, are expressed along the body axis, explain the variations in leg-bearing segments in crustaceans versus insects. A small tweak in regulation can mean legs grow on an abdominal segment or not. In other cases, similar genes might direct entirely different downstream developmental processes in various organisms, leading to their diverse body shapes even while using conserved upstream regulators.
So evolution tinkers with the controls, not just the parts themselves.
Very much so.
Hmm.
It's often about redeploying and subtly modifying existing genetic toolkits. So what does this all mean for us? The remarkable similarities we find between genomes reflect our common ancestry, profoundly linking us to all life on Earth. But it's the differences, often subtle yet impactful changes in genes and their regulation, that have driven the incredible process of evolution and created the huge diversity of organisms that exist today. It's a testament to life's adaptability and creativity.
It really puts things in perspective. Okay, so to recap, you've taken a deep dive into the genome revolution, understanding how sequencing and bioinformatics have just completely transformed our ability to read and understand life's blueprint.
A total game changer.
We've seen that genomes are incredibly diverse in size and composition, filled with fascinating non-coding DNA that acts as a vast control center, not just junk, and dynamic jumping genes that actively reshape our genetic landscape over time.
Yeah, genomes are far from static.
And we've explored how the fundamental mechanisms of genome evolution, from gene duplication and rearrangement to the constant input of mutation, have shaped the incredible diversity of life, from the smallest bacteria to complex humans like us.
That's right. And this knowledge truly matters on so many levels. From its direct application in personalized medicine and innovative cancer treatments, which we're seeing more and more of,
Right.
to revealing our deepest evolutionary ties with all living things, connecting us to the entire tree of life, and fundamentally understanding the intricate processes of development that build organisms from a single cell. We are genuinely on the brink of a new world in biology, constantly redefining what we know about life itself.
A really exciting time. So here's a final provocative thought for you to consider. Given the remarkable conservation of developmental genes, those homeotic genes, across vastly different species, what does this imply about the future possibilities, maybe the ethics of re-engineering life, or even our own species, through genetic manipulation? Something to truly mull over.
Definitely food for thought.
Thank you for joining us on this deep dive into the fascinating world of genomes and their evolution. We encourage you to keep exploring, keep questioning, and keep learning.